Hi, my name is Mariana Flores.
My name is Miriam Guerra.
And I am Ana Laura Cantu, and we're going to talk about antimicrobial resistance in bacteria.
Bacteria has been one important topic in these years. In pandemic, it has been one of them. These microorganisms have developed the ability to resist antibiotics due to the use of enzymes and alterations in the physiology. The problem with the antimicrobial resistance has arrived since years ago with a high use of not necessary antibiotics. This helps bacteria to support the effects of the antibiotic and do not get eliminated by them. Next. One example is Klebsiella pneumoniae that has a resistance to beta-lactamase antibiotics, and these microorganisms produce the enzymes beta-lactamases like Pseudomonas aeruginosa that hydrolyze the beta-lactamic ring in the antibiotic. Pseudomonas aeruginosa, it has a resistance to antibiotic by using the mechanisms in the production of biofilms. Another example is Mycobacterium sp that have a cell wall with an unusual structure that can be more resistant than other bacteria.
There are a lot of mechanisms that can help the bacteria to resist to antibiotics. Some of them are by the secretion of enzymes, proteins, DNA, or it can also be by their strong cell wall structure. These mechanisms can be a natural or an acquired resistance. Some of these take a long time to appear because of the bacteria mutations or adaptation. In the natural resistance, bacteria contain their own genes that cause some type of resistance to antibiotics and are used as a defense mechanism. In this case, all the bacteria of the same family tend to be resistant to the same antibiotic. In the acquired resistance, by bacteria undergo change or mutations in their DNA due to errors in the replication process or by transfer from other bacteria. One example is Staphylococcus aureus. These bacteria have acquired resistance to penicillin by a mutation in his gene MEK-A due to an error in their transcription process.
The relation between antimicrobial resistance and bacteriology is about the identification of bacterial pathogens, and information about their antimicrobial resistance can help in a clinical to select to appropriate medication and provide the suitable antibiotic for the treatment to control different types of infections. The bacterial mutations can be retained by sequence or acquisitions of resistance elements for another organisms. The collection of all the resistance genes have arisen by various means, like self-protection, transport, and detoxification of chemical products. And that type of metabolism demonstrates the selectivity of different types of antibiotics. As, this process can contribute to the evolution and selection of resistance. It is very important to current stocks of antibiotics and make uh, some new ones. And the major influence of resistance bacteria include antimicrobial effects and threat individuals. Some barrier techniques can help to control resistance bacteria, and all the reports agree that careful of discrimination use for antimicrobial agents for minimizing the problems.
And that's all. Thank you for your attention.